do your evaluations, at least as far as I've been informed, I only have 36 out of 60. It's
wow.
5 points. So do the evaluation, send me a screenshot of the thing that says you've completed it. Send it as an email from your SHSU email, or make sure you include your first and last name in the body of the text so I know who it's from, and include only the word marmot in the subject line. That'll automatically dump it into a folder, and sometime in the next couple days, I'll go ahead and add those to Blackboard. Okay, other things is today is sort of the last class day. A week from today, you have your final exam. Will be in this room. Not at this time, at 10:15 instead, but you'll have 2 hours to do it. It'll be the length of a normal exam, probably about 10 questions, similar to what you've seen. Okay, then we're getting ready to finish up. When we last left off, we were talking a little bit about a few questions regarding gene expression. And one of the things that was a big concern to people is sort of in the late Mendelian, before molecular genetics was just exactly what genes do. Like, what are genes actually encoding? So we're going to talk about how it was discovered that genes code for, then each gene codes for one polypeptide, and then we're going to talk a little bit about how genetics, this knowledge can be used to dissect metabolic pathways. And the reason I want to talk about that is it's about the coolest thing I've ever heard of. It's just a tremendously smart inference by this guy named Norman Horowitz. So in 1942, people knew a lot about how genes were transmitted. They knew that you could have mutations that resulted in recessive alleles, but they didn't really know how that actually worked. Okay? And George Beadle and Edward Tatum, working together at Caltech, hypothesized that the nature of a recessive allele was the result from a mutation that prevented an enzyme from functioning. Right? So as soon as an enzyme stopped functioning, then the product of that enzyme would sort of disappear from the cell, and therefore you would get a recessive phenotype of anything that was homozygous for that mutant allele. And from this, they proposed something that they call one gene, one enzyme hypothesis. We're going to talk a little bit about their experimental methods in a second, but I should let you know that this is something that we now more formally recognize as really a one gene, one polypeptide hypothesis. And that's simply because there are enzymes that are made up of multiple polypeptides, right? You can have lots of proteins that sort of work together to create the function of a single enzyme. We call those holoenzymes. But they end up winning the Nobel Prize for this in 1958. And one of the things that they did that was really smart is they found this model organism that was really, really easy to use to do controlled crosses with really large numbers, right? So they can induce mutations and find them relatively easy. And this is just bread mold. It's Neurospora crassa. We talked a little bit about its life cycle, but I want to go over it again. It's a fungus, and it's an ascomycete fungus, so it's similar to yeast. Yeast, we know, has been a microorganism for some time in genetics. And essentially, this is a yeast that spends almost its entire life, or almost the entire life cycle, in the haploid phase, right? Which is really different from us. We spend almost our whole life in the diploid phase, and animals only have a very brief haploid phase, which is basically comprised of gametes. Okay. So in any case, right, we have these haploid phases, and we can take sort of one of these spores, which is haploid. It's got one set of chromosomes, and you could grow it up in meat to form these chlamydia, which are sort of these multicellular haploid adults. And when two bread molds love each other enough, and they're in different mating classes, they can undergo cellular fusion, right? So you'll have two haploid cells that will fuse to become one cell, and the nuclei will fuse to become momentarily diploid, okay? They'll be diploid for like a second, and they immediately undergo meiosis. And this meiosis is followed by one round of mitosis and results in what's called an acospore, which is found in an ascus. An ascus is basically a sac that holds these particular spores. And the great thing about this is because they're haploids, right, we can really see what allele they actually have, because you don't have any allelic interactions in something that's haploid. There's no heterozygotes, there's no homozygotes, right? They just have what they have, one set of chromosomes. from their parents, right? And so we can use these in controlled crosses then to look at metabolic phenotypes, right? Which is kind of similar to what we were doing when we were mapping bacterial genes. The Neurospora wild type bread mold is what we call prototrophic, which means that it can grow on media that has very, very little. It means it can make most of the components that it needs for normal cell function. So it just needs a source of an inorganic salt, some nitrogen, some glucose, and some biotin, and they can grow just fine. They can make all of their own amino acids and things like that, okay? On the other hand, they learned that you could basically induce mutations using X-rays to produce these oxytrophic mutants. These are mutants that had some sort of change to an allele that prevented them from producing one of the essential nutrients that they need, right? So one amino acid or something like that. And so they couldn't grow on minimal media, but could grow on media that has been supplemented with something, right? Whatever it is that they're missing. So these mutants lack enzymes necessary for some essential step. And it was I'm using their first names for no good reason. Beadle and Tatum's thinking was that they could do these sort of crosses and they could track down things that they thought were mutants for a particular enzyme, and then they could see if they could isolate that enzyme from the prototrophic line that produced it, right, and then fail to isolate it from the auxotrophic line, which is exactly what they did. And they did a lot of experiments that followed sort of the same experimental protocol. So the first thing they would do is they would take some wild-type Neurospora and they would treat it with X-rays. Now, people didn't know exactly why this caused mutations at the time, but they knew that it definitely increased the rate that auxotrophic mutants would occur in a cell line, okay? And then what they did was they mated it with something else that was wild-type, right? So can anybody think of the reason that they would do this mating before they would isolate the ascospores that resulted? What's that?
So there's no mutation?
Well, it's not to make sure there's no mutations, but to make sure that the mutations were heritable, right? Because if you treat cells with X-rays, there are all kinds of problems, right? And so they thought like, well, if maybe the X-rays are just interfering with the enzyme, that's not good enough. We want to know that whatever change we're inducing in these lines is actually heritable. It's actually a mutant line, okay? So they would cross them with Neurospora that were wild type. They would get the fruiting bodies from them, right? And then they would dissect these acospores out of these asci, which is plural for ascus, and then they would culture each of these to see if they could detect the mutants. Now, culturing them would begin with them culturing all of these on complete media. And one of the cool things about these ascospores is it's really easy to take an ascus, dissect it, so you can open it up, and then remove a single spore and put it on control media. So you know that anything that grows from in that particular tube grew from a single ascospore. Okay, so it's all genetically identical, right? So they would grow up all of these ascuses on complete media, and of course they should all grow, even if they're auxotrophic mutants, because the media's got every imaginable thing that you could need, right? And then what they would do is they would move these to minimal media, right? The kind of media that only prototrophic things or non-mutants could grow on. And if something grew on it, they knew that there wasn't a mutant line, right? That the X-ray had failed to produce a heritable mutation that was passed on. On the other hand, if the tube was empty, right? So if this guy, this particular ascus was, or ascospore was put on here, and it was grown up, and then it couldn't grow on minimal media, we know that it's auxotrophic for something. That there's some mutation that prevents it from producing one of the essential components that cellular function requires. Okay? And so then once we have that, we can start looking at this auxotrophic line to try to figure out, well, what is it missing?
Why is there no growth on the complete medium on the far right?
Oh, it's just we're still putting them in.
Oh.
Yeah, but these are getting put in. These have already been filled. That's a good question. Yeah, we haven't put the cotton in yet. So what you do is then you take that auxotrophic line and you grow it on minimal media that's been supplemented with one thing. right? But you have a million tubes that each have one supplement added, okay? So in this case, we've got middle mediate supplemented with glycine, middle mediate supplemented with alanine, middle mediate supplemented with leucine, et cetera, right? So these are sort of all the amino acids. There are other things that they could supplement it with. And in this particular case, what they found was that the loss of function occurs in a gene that allows the synthesis of tryptophan, right? Because as soon as we add tryptophan there, that oxytrophic mutant can out. So we know that it cannot produce tryptophan. And their idea essentially was, if there's an enzyme that's necessary to synthesize tryptophan, this basically has a mutant for that enzyme. I don't know if that makes sense to you guys. These experiments were greatly improved by a guy who does not get enough credit in my book, which is why I like to talk about it. It's this guy, Norman Horowitz. Norman Horowitz was working in George Beadle's lab as a postdoc, right? So it's the best job you can really have. A postdoc is somebody who's got their PhD, so they get paid a normal amount of money, but they're responsible pretty much just for doing research. They don't have teaching responsibilities generally. You don't go to faculty meetings. You don't sit on committees, right? You just get to do science, like, for 2 years, like you were trained to before you become a professor. Then you have to do all this other garbage. Okay. So anyway, here's their postdoc. And he was thinking that he could improve these experiments and basically demonstrate particular metabolic pathways by doing these control crosses. Now, bear with me, because this is a crazy idea, but it totally works. Okay? One of the things that he noticed is that you could generate lots of oxytrophic mutants that were all oxytrophic for methionine. Okay? So none of them could survive on minimal media unless you supplement that media with methionine. And then he started thinking, well, what about the things that are responsible for making methionine? We know that methionine probably isn't just made from glucose. That doesn't make a lot of sense, right? Or any of the other sorts of organic products it has access to. He realizes that there's probably a metabolic pathway where one chemical is converted to another, which is converted to another, which is converted to another, until you end up with methionine, right? So he started thinking about what could the chemical precursors of methionine be, right? Just sort of imagining what kind of chemicals are similar to methionine, so we could supplement with those. And lo and behold, he found a number of things that were similar enough chemically to methionine that as soon as he basically put those in minimal media, this oxytrophic mutant could grow. Even though it wasn't methionine, it was something that was like methionine. And from that, he basically reasoned that he could look at all of these different oxytrophic mutants for methionine and reason his way to the order, right, that enzymes put methionine together from these different components, which is wild. In this particular example, right, we have sort of a table that shows all of these mutant strains. Now, none of these mutant strains can grow on minimal media. They need media that's supplemented with methionine. And if you supplement that minimal media with methionine, they're all good, right? So we know that they're unable to produce methionine, but we don't know why, right? We don't know what part of a hypothetical chain of metabolic events got interrupted in each one of these mutants, right? So then he basically tries to grow them on some of these other things that are similar, okay? So you notice that he's got this one oxytrophic mutant called MET8. That can't grow on any of these things but methionine, okay? So he knows that there's something in the pathway to producing methionine that prevents this from happening, and it's probably pretty close to the point where you get to methionine, right? It's maybe the last step, right? And then he notices that this other mutant, which he calls MET2, can grow if you add homocysteine, which means that it's somehow able to convert homocysteine to methionine, right? That's a hypothetical enzyme right there, right? We've got some enzyme that can maybe convert homocysteine to methionine. Okay, so he proposes this step of the metabolic pathway. I know this is a little bit hard to see, especially for poor Bianca and Jeanette, right? We have to look over the computer, right? But this basically is just an enzymatic step in which homocysteine is converted to methionine by some mystery enzyme. And then he reasons, well, what do you need to make homocysteine, right? So we have these other things. What gets converted into it? And he takes a look at some other of these mutants, and he notices that we've got one mutant, MET3, that can grow if you add methionine, It can grow if you just add homocysteine. So the homocysteine is being converted to methionine just fine. And it can grow if you add cystathionine, which means that that basically can be converted to homocysteine because it doesn't grow on any of these other things. So he proposes this step in the metabolic pathway, right? We've got the cystathionine, which is able to be converted enzymatically into homocysteine, which can be converted enzymatically into methionine. Is everyone getting the reasoning for this? Or is anybody confused about it? It's just pretty brilliant shit. Like this guy thought his way out of like a very simple experiment to something way cooler. Yeah, just the one. Yeah, he just had the one. So this tube only has cystathionine in it. And if you only have cystathionine, this mutant can suddenly make methionine, which means that these enzymes must exist in it.
So why did he think that the cystathionine could just also be directly converted to methionine instead of having to make steps?
That's a good question. So why did he think that? And the answer is I don't know. Right? I don't exactly know, other than I know at this stage people understood enzymes, right? They were able to isolate enzymes, and that was something that got a Nobel Award in 1942 for Northrop, and I don't remember the dude's name. But they were able to basically show that they could do chemical reactions from cell lysates. So they could take like a cystathionine and convert it to a methionine, right? Basically, they couldn't do it chemically, but they could do it using a cell lysate, right? Which is sort of how they hypothesized this. I think he's in this milieu, right, where people are thinking largely that there are these complicated metabolic pathways for synthesis of essential nutrients. I don't know if that's great. Yeah, he just imagined it was that way. But a lot of times in science, that's how you get to be a good scientist, is you have a lucky imagination, right? You just like, you just kind of like, you just get lucky. I mean, Crick with this, it's 3 nucleotides in a row, encoded amino acid. There's a million different ways that you could have imagined that in 1961, right? But he was like, it's got to be this way, and as it turns out, it was, right? And again, rather be lucky than good. But in any case, there's it's also a hint of good here, because like he's doing these like really sharp experiments to sort of tease this apart. So once you have the cystathionine, he notices, right, that you have this mutant, right, in which if you add O-acetyl homoserine, it suddenly can grow on that too, right, as well as all these other things. And so he reasons from that O-acetyl homoserine can be used to make cystathionine, which can be used to make homocysteine, which can be used to produce methionine. Okay, it's kind of a wild idea, but super fun. And the thing is that they took this idea and then they said, well, we're going to take each one of these lines and we're going to do all of the fractionation that's required to isolate all of the different enzymes. And we talked about this a little bit, but not in a lot of detail. This is more of a biochemistry thing. But essentially, all these enzymes have different chemical properties. They have different sizes, right? And so you can sediment them across sort of different kinds of gradients to isolate them. You can run them out on potato agar gels or potato starch gels to sort of separate them by size. There's all kinds of things you can do to sort of isolate enzymes. And what they were able to do is they were able to isolate each of the enzymes that was responsible for this metabolic pathway, okay? Which went to support sort of this one enzyme, one gene model that beetle and Tatum set up, okay? And so this is basically how they discovered these enzymes, right? They discovered that there was this homoserine transacetylase and a cystathionine gamma syntax and a cystathionase 2 cell, right? Now, one of the things I want to point out that like obsessed me about this story, right, is from what I have read, Beetle and Tatum originally were just doing this thing with Neurospora where they were finding oxytrophic mutants and they were like saying, see, we've induced a mutant which can't grow on a particular thing. That must be because there's an enzyme that's associated with that mutant that's lost its function, which I think is like sort of sad and weak in comparison. But they won a Nobel Prize for this in 1958. And the funny thing is, in their Nobel Prize address, both of them acknowledged the work of Horowitz of really improving the experiments and allowing them to make that much stronger. He's not awarded a Nobel Prize at all, right? He gets $0 from the Nobel Committee. But I mean, clearly the guy's a brilliant scientist. He goes on and has a great career anyway. But I think this is just sort of an interesting thing. I mean, this guy in no time at all was basically like, oh yeah, I'm going to basically infer metabolic pathways by doing controlled crosses with bread mold. I mean, this is like a 4-year kind of thing where he's like, he envisions it and then he's got them, which is wild. And it became a really good tool for figuring out those metabolic pathways work. A lot of people sort of cite these experiments as sort of the beginning of molecular biology, where he was one of the first people who was sort of combining genetics and biochemistry to infer really how macromolecules function together in cells.
Found these chemicals were necessary for growth, and then from there was like, okay, I know these structures of these chemicals. How do I go from homoserine to O-acetyl-homoserine and just like an enzymatic function, plugged it into that? And it's like, okay.
I couldn't say it any better. Did everybody hear what Jack said? Yeah, I mean, he just imagined what the metabolic, what the different metabolic precursors could be. And of course, I showed you like a very, very, like, you know, a perfect system. I'm sure there were lots of things that weren't part of the pathway, right? That just didn't work. But like over, you know, a period of 4 years, you figure out a number of these pathways. Yeah, it's pretty cool. Like, just, you just sat around and imagined some stuff, and then you tested it. I mean, that's really what science is, right? You sit around, you use, they call it hypothetico-deductive, but that just means like if this hypothesis is true, imagine what the world should look like. Right? That's what hypothetico-deductivism is, right? Is you use deductive reasoning to say if this hypothesis is true, what should I expect the world to look like? And that's what he did, and figured it all out, and didn't get a Nobel Prize for it. Was later admitted into the National Academy of Science. So I just want to run through something like this, and I don't know that I'm going to put a complete one of these problems on the exam because I don't know if this is good, but you might want to be able to answer questions about some of the reasoning that takes place here. Okay. So this is another example. It's just a made-up one where we have a glutamine oxytrophic strain. So it's a strain that cannot survive unless it's on minimal media that's supplemented with glutamine or one of these other precursors. Okay. And so we want to basically figure what order the supplements have to be synthesized in the metabolic pathway for glutamine. And sort of this is not such a difficult thing to do because we basically start by saying, okay, well, which one of these mutants can grow on the fewest things, right? What that means is whatever it can grow on must be the immediate precursor to glutamine, right? So if you can't grow on B, you can't grow on A, you can't grow on G, you can't grow on S, but you can grow on R, right? And you can grow on glutamine. That means that the R must be synthesized into glutamine, okay? And so that's sort of how the reasoning of this works out. So we look at mutant strain number 4. These are all, of course, glutamine oxytropes. And we say, like, okay, R is sufficient to produce glutamine in strain 4, and then we can hypothesize an enzyme that converts R into glutamine. Okay, whatever this enzyme is. Okay, then we can go from there and say, okay, well, what do we need to produce R? And we look up here in mutant strain 1, and we can see, like, if you've got B, you can survive. This mutant can survive. So that means B must be used to produce R. Okay, so adding B by itself produces glutamine, so that means that B can produce R, and that R can produce glutamine, okay? And so we've got this B to R to glutamine sort of pathway. And we just sort of keep going with this, right, to put it all together, right? So what do you guys think is next? What do we need to produce B? Yeah, right, T, right? Right, so adding T by itself produces glutamine in strain 3, and so we can say T is needed to produce B in some enzymatic reaction, which is used to produce R in some enzymatic reaction, which is used to produce glutamine, right, etc. Now, you get asked also what's used to produce T. I can't believe I worked this through all this way. Hey, yeah, that's sort of easy, right? So this can basically grow on any one of these things, which means it's the farthest from glutamine, so that basically A can produce T, can be converted to T by some enzyme, T can be converted to B by some enzyme, B can be converted to R by some enzyme, and R can be converted to glutamine by some enzyme. And there we go. We've got a metabolic pathway, and now we can use these mutant strains to start to look for those enzymes themselves, right? And once you basically start doing that in fractionated enzymes, all you need to do is say, like, okay, well, I'm going to add this particular fraction to a tube that has A in it, and let's see if I can make T. Right? That's pretty much it. So it's a mix of sort of biochemistry and genetics has basically produced this field that we call molecular biology. Does it make sense to everybody? And of course, S doesn't produce anything that's not in the pathway. S doesn't help. And so I like this example just because it shows that like there were probably things when Horowitz was working this out where it's like, well, this clearly isn't part of the pathway because I can't find an auxotrophic mutant that can grow with the addition of it. So a few other things that I want to add before we move on and talk about structure of proteins is, of course, the fact that not all polypeptides are enzymes. Okay? In fact, some of the most common polypeptides that we have in our bodies aren't enzymatic at all. They tend to be structural, right? So I don't know what the most common polypeptide in humans is, but I would guess it's collagen, right? Collagen is basically used in bones and connective tissue and muscle and stuff like that. You have it all over the place, right? It's structural. It doesn't actually do any chemical reactions. An example of one protein that was discovered very early on that doesn't do chemical reactions either is hemoglobin. And this is discovered based on studies of a genetic disease which is called sickle cell anemia. And this is something that basically is a disease that can cause what's called sickling of red blood cells. So the red blood cells somehow suck in at the middle and they sort of get the sickle shape, okay? And that makes them worse at carrying oxygen, right? If they're sickling, they're not carrying oxygen so well, right? And so that results in all kinds of physiological problems because it probably is obvious to you guys that you need oxygen kind of all the time, okay? So low oxygen means that you have low energy. Sometimes you're more likely to get sick, right? People have a tendency to feel unwell. They turn a little bit blue, whatever, okay? So this is a problem, and it's a recessive genetic disease. So if you are wild type for both of these hemoglobin genes and mutations, you have normal functioning hemoglobin, you don't get any sickling. If you're heterozygous, you're what is called somewhat anemic, right? Which means that you do have some sickling take place, but it's not enough that it's extremely dangerous. It's just hella inconvenient, okay? And then, of course, if you've got a recessive homozygote, that person is dangerously anemic, probably doesn't end up surviving. Without talking much more about this, does anybody know what disease sickle cell anemia is most closely associated with as like sort of prevention? Yeah, well, actually, yeah. Being a heterozygote is selected for in areas with high malarial concentrations, and that's because amoeba don't do well in blood with a lot of sickling. It makes it hard for them to get around, okay, which is kind of wild, right? So this is a disease that actually is selectively advantageous in some environments. In any case, in 1949, Linus Pauling had sort of already come up with the structure for hemoglobin, and in this he was studying it to basically explain how sickle cell anemia works. And this is how we detected two electrophoretic variants of hemoglobin in sickle cell heterozygotes. Essentially what he found was that if you look at hemoglobin, it's sort of it's a bunch of polypeptides that are sort of bound together. We've got two alpha subunits, which are shown in blue, two beta subunits, which are shown in red, and then you've got four of these heme groups. A heme group is just this weird carbon lattice, and at the center of it holds some usually metal ion. Right, so you would find that also in chlorophyll. Chlorophyll's got a manganese in there. Does anybody know what metal ion is in the middle of hemoglobin?
Iron.
Iron, yeah, okay. It gives it its red color, right? So when you oxidize that iron, the iron is actually carrying the oxygen to different parts of the body. But in any case, what they found out is that this sickle cell recessive allele was a result of a single substitution. Right? You have one mutation that results in the production of a glutamine, a valine at amino acid 6 of the beta subunit, right? And that changes the shape of that subunit a little so that when you don't have very much oxygen, rather than, of course, binding oxygen, that subunit will bind other alpha subunits in hemoglobin that is not itself, right? So what you'll have is these hemoglobin molecules that will stack up on top of each other, right? And so none of them can bind oxygen now, and them stacking up like that causes these fibers to occur inside of these erythrocytes, and that's what causes them to have that sickle shape, right? It kind of sucks them in the middle. Okay, so you have these adjacent, and I shouldn't call them holoenzymes because I've just said not every protein is an enzyme, but I'm just so used to using the term holoenzyme, right? They bind each other, and this abnormal shape results in this change. And this is also sort of support for this idea that one gene encodes one peptide, because when you mutate one gene and the result is you change one polypeptide, it supports that hypothesis. We're almost at an end. And where we're going to finish is actually mostly way easier, because we haven't finished talking about it. It's sort of the end of chapter 13. What's the point of all of this, right? We've talked about transcription and translation. The point is to produce proteins that do particular jobs in cells, right? Proteins are really the workers of the cell. They do chemical reactions, they have structural jobs, things like that, okay? And so I want to spend sort of the last little bit just talking about what proteins are and how those work. And some of this, I think, is probably going to be reviewed for most people. Proteins, as you guys know, are also called polypeptides, right? And they are made up of chains of chemicals which are called amino acids. Amino acids have a common structure in that they have a central carbon, which is called the alpha carbon. They have two functional groups on either side of the central carbon. One of them is an amino group. One of them is a carboxyl group, right? And this is a positively charged group. This is a negatively charged group. And they all have something called an R group that also is attached to this alpha carbon. And the R group differs between amino acids. That's what gives the different amino acids the different characteristics. And the idea is, of course, we can link up different amino acids by connecting carboxyl groups to amino groups. They're a little bit like Legos in that regard. And we call that connection a polypeptide or a peptide bond. And the way that it works is easy. It's a condensation reaction, if I recall correctly, right? So we've got this carboxyl end, right, which can unite with a amino group, and it releases two H's and an O, which essentially form oxygen or water really quickly, because oxygen hates to be alone. It'll bind up any hydrogens nearby. And so then we end up with a bond that basically looks like this. We've got a nitrogen that's connected to a carbon here, right? And so this is what we call a peptide bond. And if you've got a bunch of these in one molecule, we call that a polypeptide. This is real basic review, I hope, right? And there are lots of different amino acids. And there are like thousands and thousands of different amino acids, but all living things only use 20 of them. Okay, and this is the 20 things that go into organic proteins or organic polypeptides. And they all have different sort of chemical characteristics. So we've got a number of these which are neutral. They don't really carry much charge. We've got some non-polar ones, which are also so neutral. We've got some basic ones, and we've got some acidic ones, right? Sort of these all function, they interact together to produce the qualities of a polypeptide, right? We get all these amino acids, and the interactions of those R groups are what basically cause a polypeptide to behave the way that it does, okay? And there are really 4 levels of structuring that we talk about when we talk about polypeptides. That, again, there's some people talking about MCATs before this starts. You should just know these. You're going to take them, okay? So it's kind of important to discuss them. Okay, and we can think about this a little bit like we thought about the structures of DNA in eukaryotes. Right, we start with sort of this 3 nanometer fiber, where it's just you have DNA wrapped around a single nucleosome, and then those nucleosomes sort of spiral around each other, called that 11 nanometer fiber, right, et cetera. This is sort of the same idea. Okay, the first level of organization is just what we call the primary structure, and this is the order of amino acids in a polypeptide chain. You guys did some stuff with your lab where you had to do some blasting, if I'm not mistaken, right? You had to find things. And if you read down through the way that large polygene areas are described, you often see stuff on your NCBI thing that look like this. So we've got a large subunit of something here. Oh, I know exactly what this is by this sequence. This is ribisco. It's my favorite one. I'll talk about that in a second, right? And this is basically the amino acid that's encoded by the DNA, right? That sort of will show up in your nucleotide blasts, okay? And that's all that the primary structure is ordered, okay? But when we realize that R groups will interact with each other, and they're most likely going to interact with other adjacent R groups. This produces sort of higher level structures that we call secondary structures. Secondary structures are sometimes defined as sort of the regular folding of polypeptide chains based on interactions between adjacent R groups of amino acids, right? So if you get a bunch of amino acids that are sort of close together, they can sort of form these higher level structures, right? One of the first ones to be discovered was the alpha helix, which was discovered by Linus Pauling, which is a result between interactions of R groups of adjacent things to sort of form these spirals, right? Or in some ways, they're like rods or tubes. But we also have sometimes polypeptides which can form sheets, right? So they can form sort of these flat surfaces by essentially strings winding back and forth and R groups interacting with each other. And they're sometimes called pleated sheets because they have a tendency to be a little bit corrugated, okay? That is just part of the structure of a protein, right? It's having these alpha helices or beta pleated sheets. You also have what's called tertiary structure, which is the structure of an entire protein involving all of these parts sort of working together. And often this involves interactions between R groups that are not adjacent. Okay, so here's an example of one such protein sort of been stylized this way. We can see that it's got a couple of these helices, it's got a couple of sheets in it, and these are all interacting to produce this higher order structure, which we sometimes refer to as the conformation of a protein. It's the way that the protein basically is structured in its entirety. This depends on lots and lots of different kinds of interactions between R groups, right? Some of these interactions are really, really strong, right? So we've got disulfide bridging, for example. That's a huge one in proteins. There's only 2 amino acids that have sulfur in them. Does anybody know what those are? Cysteine and methionine. Studying for the MCAT. Yeah, cysteine and methionine, but they can form these covalent bonds which hold a protein together really, really strongly. There are also weaker bonds that can exist between non-adjacent R groups of amino acids, like you get hydrogen bonding taking place. Sometimes ionic interactions because we've got polar and non-polar amino acids that will change that shape somewhat. And also van der Waals forces. Now, every time this pops up, I have to look up what those are, and I'll be honest, I still don't really get it. Like, I know that it's very, very weak forces that have to do with very, very weak charges that vacillate inside of atoms and molecules. That's where I am on that. So that's it. That's Ask van der Waals. You've got questions, you can Google yourself. Clearly not going to be any questions on the exam about that, because I have to write a key. And then, of course, right, we have a higher order structure, and this is the last one, which is quaternary structures. We know that proteins, and certainly when we started talking about replication and things like that, you get these big complexes, which are lots and lots of proteins working together, okay? So the structure of these complexes is what we call quaternary structure, right? An arrangement of multiple polypeptides into a single multi-subunit complex. So this is basically a picture of one of these complexes where each polypeptide has a different color. Okay, so you get those arrangements, and those can become important because sometimes the way that different polypeptides join together change the conformation of individual polypeptides and makes them enzymatically capable, where when they're alone, they're not, right? It sort of changes their ability to do enzymatic reactions, right? And I want to talk about my favorite protein in just a second because it's got this really exciting quaternary structure that allows it to work. And the other thing is that this is the most common enzyme on the planet. So people should know this. And the reason I'm talking about it is I've worked a lot on the evolution of this particular protein in parasitic plants for my dissertation. Ribisco is also called ribulose 1,5-bisphosphate carboxylase oxygenase. And this is the enzyme that takes carbon dioxide out of the air and basically adds it to a molecule of ribulose bisphosphate, which is basically what all of us are made of. Every carbon molecule in all of our bodies came out of the air through this molecule, which is crazy when you think about it, okay? But in any case, it's what's called a hexadecamer. So it's massive, and it's made up of 16 different proteins, okay, of two types. There are 8 large proteins. The large subunit is coated in the chloroplast, and it actually has enzymatic activity, right? And if we look, the large subunits actually sort of form this barrel shape. So we've got the red and green are large subunits shown here. We can see them from on top over here. And then we have 8 small subunits. One of the fun things is that the small subunits are encoded in the nuclear genome and have to be imported into the chloroplast. And it's the importation into the chloroplast of the small subunit that regulates expression of the large subunit. Right? How cool is that? You got these 2 genetic compartments that are working together to produce something which is really, really important. Now, the small subunits don't actually have any enzymatic activity at all. But when they bind to large subunits, they change the conformation of those large subunits enough so that the large subunit becomes 100 times more efficient at binding carbon than when it is not bound. And again, it's the most prevalent holoenzyme on the planet. 40% of leaf protein. And if you guys go outside, there's a lot of leaves out there. So I do want to talk a little bit just about how proteins sometimes also find their shape, because it's not 100%, random interactions with R groups that gets them to the right tertiary and quaternary structure. Sometimes they need help. And they need help by other proteins which are called chaperones. Chaperones are proteins that basically are associated with ribosomes, and as polypeptides come out, they fold them the correct way. There are a number of these that are important for protein folding, and they have really easy to remember names, which is nice. You have things that are called foldases, right? And those are ATP dependent, so it requires energy, but they fold proteins into less probable conformations. That's why it takes a little bit of energy. Then there are holdases, which bind folding intermediates to prevent aggregation. And then there are disaggregases, and what those basically are doing is they're preventing partially folded proteins from binding other proteins that they don't want to bind to, right? That is important to keep them from binding to, okay? So they basically pull inappropriately assembled protein complexes apart, right? And keep that from happening. And finally, there's a kind of chaperone called a chaperonin, and these are basically mostly activated in response to heat shock. And that's simply because when you change the temperature of a cell, you change sort of the interactions that exist between amino acids and polypeptides, right? And so they have a tendency to fold incorrectly, or sometimes that would even denation, right? Raising the temperature of a cell sometimes can cause hydrogen bonds to loosen and things like that. You guys already saw that with DNA, right? You guys did PCR. How did you denature the DNA? You made it hot. That's all it takes, right, to break up hydrogen bonds, right? And so chaperones basically are activated when a cell is in heat shock to sort of help keep proteins folded until that stress is actually over. The last little bit I want to talk about, and I don't know why I have all of these animations on the last slide, I apologize for that, is misfolding and aggregation of proteins as a basis of human disease. This is a really exciting thing that people discovered in my lifetime and when I was old enough to kind of follow it. But there are a series of diseases which are called prion diseases. And prion diseases essentially result from proteins that are misfolded. But not only that, but if you have a misfolded protein, that protein will now function to misfold other proteins, right? And that can be a real problem, right? So you have one protein that's misfolded, it finds other proteins that are like it and unfolds them, okay? And there are a number of different pathologies that we know of, at least in humans, that result from a single misfolded prion protein, which is called a prion protein, or PR. And this is a protein that is associated with neurofibrils in the brain. And what happens when these become unfolded or folded incorrectly, right? They basically will spread that wrong conformation to adjacent proteins, which will happen. And over time, you'll develop all kinds of neurodegenerative symptoms, right? And there are different kinds of diseases that result from this. The most famous one is probably one that's called Creutzfeldt-Jakob disease. Sometimes they also call it mad cow, or when you get it in sheep, it's called scrapie. And as it turns out, the neurofibril proteins in cows and sheep are similar enough to ours that if you consume those proteins, you can get the disease, right? I don't know if you guys remember this because you would have been very little, most of you, but there was a mad cow outbreak in the UK in the early 2000s, late 1990s, and essentially they had to murder all these cows, right? They just had to put them all down. And they found out that the reason is that they were using actually weird beef parts in the food that they were feeding the cows, which is kind of sick and weird when you think about it, right? And some of what they were using was brain tissue, right? So they were feeding cows parts of the other dead cows that nobody wanted to eat, like the brains and stuff like that. And so you would end up with sort of these, these cows all sort of having these neurodegenerative problems, right? Mad cow disease basically results in the cows behaving really erratically. They call it scrapie in sheep because they will scratch themselves almost to death. And Jacob's Creutzfeldt disease in humans, I think the first symptom, if I recall correctly, is like you just can't sleep, right? It results in like this really chronic insomnia. And as it turns out, people who can't sleep eventually die of that, right? And it's horrible, right? Like because like after a couple days, everything that you see is like kind of a mix of reality and hallucination, really, really hard on people who get it. It's also been associated with a disease which is called Kuru, which is really, really rare, but is sometimes occurred in the peoples of Papua New Guinea, especially tribes that were cannibalistic, right? So it was regarded as sort of like, and I don't like know the full anthropology of this, is like if you defeated an enemy, like somehow you could obtain that enemy's strength by devouring your enemy, right? And you just devour the whole thing, you eat some of those brain proteins. If he's got a little misfolding in there, right, all of a sudden you develop this, and it's got similar sort of neuropathological symptoms as Creutzfeldt-Jakob disease. That's like if you guys are interested in Googling weird stuff, I mean, these prion diseases are super weird because they are transmissible, but there's no virus involved, right? There's no heritable nucleic acid involved. It's just proteins, and they just go around and misfold in the protein. That's all I have. So this is sort of just a summary for chapter 13. It was a long one. We talked about protein synthesis and how it requires the interaction of many RNA and protein components. For the exam, you should definitely make sure that you know about what those components are, including RNAs and ribosomes, but also tRNAs and the amino acid tRNA synthetases that charge them, as well as mRNAs. Okay? We talked in a previous chapter about how those mRNAs are produced. We talked about protein synthesis requiring 20 steps for initiation. I forgot to fix the slides before I put them up, so it looks like there are 22 steps. They're really only 20, maybe even less. I don't know. There might be other mistakes in there. Right, but you should have an idea of those, and I've included in your review a question that I think you should pay attention to because it indicates what parts I'm specifically thinking you should know about. So it's like basically like, here are the players, tell me the story of how translation actually works. Unlike transcription, prokaryotes and eukaryotes are really, really similar in the way that translation happens. Okay? And then today we talked a little bit about Beadle-Tatum's experiments, which were greatly amplified by Horowitz to basically show how one gene encodes one polypeptide, okay, and protein structure. So that's all you need to know for your exam. Does anybody have any questions? Okay, one thing that I want to point out is that I know that tomorrow is a reading day. I will be in my office during office hours. In the morning, if you can't make it, let me know. You can come to my office and read it another time of the day. You can figure something out. And I'm going to basically treat next week more or less the same. I'll be in my office most days from or my lab from 9 to 5-ish, somewhere in there. So if you have questions, you want to stop by, shoot me an email, and we can kind of help you get your stuff worked out. Logan.
Do we need a Scantron?
No Scantron. It's going to be just like the other ones. Which means that the weekend after this is going to be horrible for me because the amount of grading I have. Because I got to get those grades on Monday, so I have Friday, Saturday, and Sunday to get your stuff graded.
Do we need a calculator?
Do you need a calculator? It wouldn't hurt to bring one, but I can't think of any problems that'll need them because we're done with mapping, and that was the big one. But if you have a calculator, it won't hurt. I haven't written the exam yet, so I'm planning on doing that tomorrow morning. All right, I'll see you in 1 week, and have a good week. Study like crazy.